All right, why don't we get started? I see people are still trickling in, but I will get things rolling. I will be moderating the last two sessions, and I'm really excited to hear from our graduate student population in the LTER in the next session. So graduate students, are really an integral part of the research and education mission of the entire LTER program. And they're also a challenging population to sort of keep organized and wrangle because, they keep graduating and moving on to amazing careers. And there's always new students starting. And for our site, I don't know if this is a bigger challenge than at most. Our graduate students are spread among many institutions in New England. So having each LTER site has a graduate student representative. And about a year ago, we convinced Luca Morreale, who's a graduate student at Boston University, to take on that role at the Harvard Forest LTER. And we're really glad he did say yes to that because he's done a lot to really bring the graduate student community more together, and he'll be talking a little bit about that. But first, I have to tell a funny story. Don't worry, I'm not going to embarrass you, but it relates to your research. So at the break, I went outside for a few minutes and ran into my next door neighbor, who we share a hedgerow on either side of our farms. And she was like, "Can you come look at this tree that just fell over on the edge? I've been noticing, I think all these trees are falling over on the edge more than inside the forest." And I was like, "Oh no." First of all, she didn't know what it was. And so I got to identify it and feel smart. But then I got to say, I just went to a talk that showed that not only do edge trees in this region grow faster, but they're not more likely to die. And she was like, "Okay, cool." So science translation in action. Thanks for your excellent research and your excellent leadership of the graduate students. So take it away, Luca.
Thank you for that wonderful introduction, Audrey. Though your story does make me, I know it's anecdotal evidence, but does make me scared as I study edges, and we are asserting that trees don't die more on the edge. So, maybe more work to be done there. I also want to note, Clarisse just posted in the chat, and it is important to acknowledge that I am by no means the first graduate site representative, and everything I am trying to do and we are trying to do is building on a really excellent lineage of graduate representatives. So I thank everyone that's come before me, and thank Audrey and everyone that I get to work with here at the Harvard Forest. With that, I would like to jump in and share what we have been doing in the past year and what we're trying to do in the year coming. Can everyone see my slides? Great. As Audrey mentioned, I am a PhD candidate at Boston University in the Department of Earth and Environment, working with Lucy Hutira. And before that, I actually worked at the Harvard Forest and worked with Jonathan Thompson, and still get to work closely with his lab. And I'm always happy to be, quote, back at the Harvard Forest, even if virtually, to see so many familiar and friendly faces. So, as Audrey mentioned, graduate students are challenging as a community, not because of any particular thing. They are wonderful as a whole. I say this with no bias, being one. But there's lots of transitions. They come from different institutions and they work on different things, and what exactly they work on can actually change over their tenure as a graduate student. And so very briefly, I just wanted to answer a question that we've gotten a few times. Who counts as an LTER graduate student at Harvard Forest? Particularly, as you will hear in a little bit, as we are happy to announce that we get to offer research awards now to graduate students. Who is it that is eligible? Who counts as a graduate student? And the answer is that we take a very expansive view on this. Anyone whose PI is associated with the Harvard Forest does work here, or if the graduate student themselves does work here, obviously counts. But we also are happy to be inclusive and welcome people that, as long as it fits in with their graduate studies and fits with their research interests and their advisors are okay with it, even if they have not done work before at the Harvard Forest, we welcome them to reach out and to join the wonderful group of people we have here. In addition to the challenges that are perhaps posed by coordinated graduate community, I just want to highlight that there are actually a lot of unique opportunities that come from an LTER and come from long-term research at Harvard Forest for a graduate student, and things that I wasn't necessarily aware of before I became a graduate student and have started thinking along these. And so these include that you automatically get an institution beyond just your home institution to connect with, to network with, to learn about research that can connect to yours and is maybe on the same system as yours, same general questions, but is coming from very different angles. And as a growing graduate student myself, it is really, really, I'll just say awesome to be able to come and see all these things and just be exposed to research about, even though I study trees, right? But I get to hear about soils and get to hear about all sorts of things and get connected to the schoolyard LTER program and just all of these things that might not be available in a home department or might be available, but it's just a whole nother avenue of that. And then to the long-term research itself, a lot of times when trying to figure out our graduate path through, we're trying to figure out what questions can we ask, what are we interested in, but also what do we have time and money for. As a as an average PhD student with maybe 5 or 6 years, you have a limited amount of time to go out and collect data yourself, or be in the lab collecting data, or for me, be on the computer processing data. And so the opportunities provided by Harvard Forest and the LTR in general are just a really great way to get integrated into a broader science network and also become aware of that maybe you don't need to go out and collect that data. Maybe that you can access 20, 30 years of data that is on your same research site or on your same research question. And so as a graduate student, it's just really exciting, and it is part of what motivates me to accept this position and do it, is that I think I just want more and more graduate students to realize that in our backyards, so to speak, in New England, that we have this availability of data and research questions that could improve and enhance our own research studies. And so given that we have all of these things, we want to build on them and continue, the efforts by Harvard Forest to include graduate students, and we want to figure out how we do that. Particularly in the time of COVID, as so many things are, that can be somewhat challenging. Very few of us are at Harvard Forest. In general, graduate students are not stationed at Harvard Forest throughout the year, in my experience. And so they may be out there in the summer doing research or advising REUs, but right now and in the past year, that has been a little bit challenging. So what is it that we've been doing and thinking about doing? One piece of this has been to start a new graduate newsletter where we share events at Harvard Forest and spotlight research by graduate students. Our first ever newsletter went out just a few weeks ago to advertise the symposium and also highlight our first ever research award winners, which we're going to hear more about and hear from in just a few minutes. And so we will continue this, not on necessarily a set interval, but to celebrate events. If anyone has any ideas or wants to have their work recognized or highlighted, please reach out to me or to Jonathan or Audrey, though they will probably forward it to me. And we will gladly do so. In addition, we have an opportunity that's actually just coming up next week, which is, as Harvard Forest is just one of the LTER sites, there are also a lot of initiatives by the LTER network as a whole to foster inter-site collaborations, but also highlight graduate research in general, and more specifically. To that end, the graduate committee for the LTER, which is made up of the representatives from each of the 28 sites, has taken over the LTER Instagram account and is rotating through sites to highlight the work they do there and show the cool pictures of their work. And so the Harvard Forest Spotlight is actually coming up next week and will run for 2 weeks. So please send me pictures. I've included a sample of some of the photos from just the last few weeks. And as you see, I think we might have some competition. I personally don't have any pictures of, I think that is a humpback whale, though I'm by no means cetacean expert. So I would love any pictures you have, especially from graduate students or showing graduate students in the field, to join in with the LTR community in celebrating our network. Speaking of the LTR community, one of the things that we would like to do in the coming year is take advantage of a lot of the network level engagement. As I mentioned, the LTR network as a whole is also focusing on and trying to bolster the graduate community, and also trying to encourage sites to work cross-site. And they have money available for this, and they are trying to work to create groups and grants. And they have explicitly said that graduate students can lead these groups, and that we can try and set up a cross-site synthesis and connect over research, or even produce a product with graduate students and researchers at other sites. There's also an all scientists meeting that occurs every 2 years, I believe, where there is funding, 3 years, thank you, Audrey. And there is funding for graduate students, I'm not sure exactly how many from each site, but graduate students to attend and engage further with the LTER. And so these are things that you will be hearing more about, but also if you are interested in any way, particularly the cross-site synthesis groups or writing grants, that we would love to take advantage of and love to have a kind of Harvard Forest run, or spurred by Harvard Forest, to connect with maybe the other forested sites. Or if you can come up, if you have ideas for how to ask similar questions to the oceanic sites or any other ecosystem, that would be awesome, and are things that we are looking at. One of the bigger things, which is very exciting to announce, that we have started in the past year is that last fall, we sent out a call for applications for the inaugural Harvard Forest LTER Graduate Student Research Award, which is a little bit of a mouthful. And honestly, Jonathan and Audrey, we should probably come up with a shorter name for it, but it tells you what it is. And so we sent out a call for applications, and they were selected obviously by quality of the research proposal, but also in part on the potential of their proposals to leverage extant Harvard Forest LTER research, kind of what I was saying before. We have this wealth of data available and questions that have been asked before. And so we were looking for people to take advantage of that. And then also to create new within site collaborations. At the fall symposium, the delayed 2020 symposium, associated was the, I think, annual PI meeting, which graduate students were invited to hear about kind of elevator pitches from each of the co-Is about how their work fits in at Harvard Forest and opportunities for engagement. And so that it was really exciting to see the applications we got. And then we were able to announce the award winners at the end of the fall. And we were actually going to hear from all of those award winners and their projects in just a minute. So hold on for that. And finally, we are thrilled to announce that we are going to get to do another round of awards for this spring, and with a deadline in mid to late April, and we will be sending out an official call for that soon. And we'll be looking for similar things, people who are, and again, going back to the kind of definition of who is an LTER graduate student, this is a great opportunity for, if you haven't worked at Harvard Forest or are looking to be more involved at Harvard Forest, this is a way to, look at the cool research that is already here and come up with a research project that maybe melds your current research with the other stuff going on, or anything really. We're really excited to announce this and excited that we've already been able to award some. And so with that, I am going to stop talking and hand it over to our award winners, our 4 award winners from the fall. And it's my pleasure to introduce them. The first award winner, unfortunately, could not make it due to teaching responsibilities, one of the many things that graduate students get to do and balance, but has kindly sent in a pre-recorded elevator pitch. So, oh, actually, I just remembered I need to share the sound. Thank you for that tip, Audrey. So it is my pleasure to introduce Thomas Muratore, who is a PhD student at the University of New Hampshire, and we're going to hear about his proposed research and his current research.
Hello, everyone. My name is Thomas Morreale, and I am a PhD student at the University of New Hampshire. Today, I'll be sharing with you all my work focused on partitioning root versus mycorrhizal-derived soil organic carbon. Soil organic carbon represents a large reservoir of global carbon, and globally, soils store over 2 times the amount of carbon in the atmosphere and vegetation combined. In addition to storing carbon, many terrestrial ecosystem functions are underpinned by soil organic carbon, such as retaining water and nutrients for plant growth. Despite the importance of soil organic carbon, we lack a good understanding of the origin and formation of stable organic matter. Significant evidence indicates that below-ground carbon is a major source of soil organic carbon, with data showing that roots and their mycorrhizal counterparts influence soil organic carbon storage and decomposition differently depending on the environment. Recently, a variety of root mycorrhizal characteristics were identified as positively impacting soil carbon stabilization. The biomass, production, and turnover of the fine root system indicate something about the quantity of carbon that's entering the soil environment. And within the fine root system exists a variety of traits that significantly influence soil organic carbon storage in the order of 10 to hundreds of years. There are biological associations with mycorrhizal fungi which have been positively associated with soil carbon formation and destabilization. In addition, the chemical composition of roots and their physiological capacity indicate something about the function of the fine root system and the ability to form soil carbon. Thus, root characteristics may change in a way that alters soil carbon stocks. The objective of this work is to examine how roots, mycorrhizal fungi, and the soil environment interact to influence below-ground plant-derived carbon in a global change context. So far, I've collected measurements from a spectrum of environmental change gradients for root biomass and mycorrhizal colonization to get a sense of the do-it-yourself nutrient economy or the outsourcing nutrient economy to mycorrhizal fungi within the fine root system. On the x-axis of both figures, I show treatments for control, soil heating, nitrogen addition to the soil, and their interaction. On this first figure, on the y-axis, I am expressing root biomass in grams per meter squared. So what I found is that soil warming has reduced root biomass, with this most significant reduction in root biomass occurring in the heated by nitrogen plots. However, this is not the full story. If we look at mycorrhizal root colonization, we see that heating and nitrogen addition significantly reduced the amount of root tips that were colonized by mycorrhizal fungi. However, when heating and nitrogen were applied together, we see that there is no significant difference from the control, indicating that there is a significant interaction between heating and nitrogen addition. The next step is to examine the relative contribution of root versus mycorrhizal fungi and soil carbon inputs in response to warming, nitrogen addition, and their interaction. I will achieve this by using the stable isotope ingrowth core method, which the ingrowth core that I propose on using is seen here, where a rigid plastic mesh that has a size diameter of 2 millimeters will be used to allow the ingrowth of roots and mycorrhizal fungi. The inner core will be a hyphae-only ingrowth core with a 37 micrometer mesh that will only allow the ingrowth of fungal mycelium. I have gathered soil from a long-term continuous garden agricultural field that has soil organic carbon with a delta 13C signature that is different than the plant carbon that will be entering these cores at Harvard Forest. This will allow me to use a mixing model to then determine the fraction of root or hyphae-derived carbon that is entering into these ingrowth cores. This experiment will take place at the soil warming by nitrogen addition experiment at Harvard Forest. This study will begin this summer and will continue for 2 growing seasons. I look forward to sharing with you all the results from this study as I begin to determine the effect of root versus mycorrhizal derived soil organic carbon in global change context.
Great. Oh, I'm sorry. Were there challenges with the audio? Apologies.
A little, but it wasn't too
Thank you very
terrible.
much. Great. Thank you. Well, with that, thanks to Thomas, and it is my pleasure to announce our next award winner, who's a PhD student at UMass Amherst, and hand it over to Amanda Suzzi.
Good afternoon, everyone. So I'm Amanda Suzzi. As he said, I am a graduate student at the University of Massachusetts at Amherst. It is my 3rd year as a graduate student. And with Harvard Forest, I am working to build global change driver data literacy among K-12 teachers and students. So I'm working with Pamela Snell and Clarisse Hart to broaden, oh, let me put chat on. Oh, thanks, Clarisse. To help underrepresented groups in STEM learn more about global change drivers, climate change drivers. You can see as I'm speaking, the land acknowledgement that we are developing to add to the data nuggets. So while my dissertation is actually primarily focused on spatial statistics, a lot of my research is at the intersection between environmental justice and social change. So for my dissertation, I'm looking at lemurs in Madagascar and better ways to conserve them. So a lot of that has to deal with these underrepresented minorities and how we can adapt conservation strategies so they don't negatively influence these groups. And I'm bringing that back to Harvard Forest by including things like a land acknowledgement into the science for the schoolyard ecology data nuggets and production. We're also creating a slideshow that relates water resources to social economic status. Please excuse me, someone's at the door. Good timing. So we're making sure that there's diversity in the data nugget slideshows. We're making sure that it's not just race-based, but income and sexual status, gender, that there are different intersectionalities between the diversity we're including in the slideshows. I'm also helping build a lesson plan on the NELF Explorer so that it's a little easier for K through 12 students to understand. I'm providing some teacher mentorship support around some easy statistics and graphing skills. And I'm also in the future gathering data sets for a new data nugget with diversity, equity, and inclusion resources, possibly around carbon, although we haven't nailed down the details. Luca, would you like me to take questions now or wait till the end?
If we can take questions at the end, I'm actually not exactly sure how we will be on time. So if We'll see if we have time at the end. Thank you, Amanda.
Cool, you're welcome.
Thanks a lot for that, Amanda. And now I'd like to turn it over to Nikhil Chari, who's a graduate student at Harvard University.
Hi, everybody. Let me get this in presenter mode. Can everyone see my thing? Okay, great. So thank you. First of all, I just want to say it's just extremely exciting to be able to present here in front of all of these amazing scientists at the Harvard Forest. And thanks a lot to Luca for organizing this and to everyone for their amazing presentations so far today and yesterday. So I'm just going to talk really quickly a little bit about root exudation and its relationship with soil organic matter. So the question that kind of motivates me here is, how do root exudates interact with mineral associated organic matter, or MAOM? So MAOM is supposedly the longest cycling fraction of soil organic matter, and it's basically plant or microbial derived carbon compounds that are absorbed directly somehow weakly or strongly to soil minerals. And root exudates are basically these other molecular compounds that are released from plant roots. And these can be things like simple sugars, amino acids, organic acids, etc. And the amount and concentration composition of root exudation is projected to change with elevated CO2. So this is simply because, you have more CO2 in the atmosphere, plant takes up more carbon and allocates it in different ways. So I'm really interested in the ways that different root exudate composition can lead to MAOM formation or loss. So to this end, we've developed an experiment where we have these three different root exudate treatments. So we have glucose, which is simple sugar, which is really bioenergetically favorable. We expect this is going to get taken up by microbes, and then part of it is going to be respired, and theoretically part of it should form microbial biomass and then form MAOM through the microbial assimilation pathway. Then we have these other organic acid, which is succinic acid, and aspartic acid, which is an amino acid, and these are not bioenergetically favorable. So microbes don't want to take these up. So we kind of think the OH groups on the ends of these acids are going to go and kind of bind directly to soil minerals, and in doing so, kick off native mayom, which could potentially lead to a net carbon loss. So then the big question mark here is the reactivity of the amino group on aspartic acid. So as you can see, we've purposefully picked functionally analogous organic and amino acids so that we can really assay the reactivity of that amino group. So is that amino group behaving like a OH group, hydroxyl group, and binding to a mineral and kicking off native carbon, or is it potentially alleviating a microbial nitrogen limitation, for example? So we don't know, but we've developed this really cool experiment to test this hypothesis. So basically, we've gone out to the Harvard forest last Thursday and sampled intact soil cores, a bunch of them from right outside the control plot at the Barwoods site. And we've taken these soil cores back to our lab at the Arnold Arboretum and assembled this really crazy organism here in the lab, taking up our whole bench space. So in the middle here, you can see these blue falcon tubes, and in each falcon tube is an intact soil core. And connected in each intact soil core is an artificial root. And each artificial root is connected to one of these syringes, and then each rack of the syringes is filled with a different root exudate solution. So we have those three I talked about earlier. There's 2 concentrations of each, and then we also have controls. And so this experiment, it started on Friday. It's going to run for 30 days. And then at the end, we're measuring all kinds of things. We're measuring all the different soil fractions, carbon and nitrogen. We have 13C isotopes in the exudate solution. So we're tracing 13C. We're measuring metals in the pore water. We're measuring microbial biomass. And so I'm really excited about what we might find from this experiment. And I also think this setup is super cool and could potentially lead to several future experiments. Among them, we've taken a Harvard Forest REU student this summer who is going to propose an experiment on this setup. And I'm just really happy to hear anyone's comments or questions or ideas in the chat if you have any. So thanks a lot. And that's my presentation. Stop my share.
Thanks for that, Nikhil. Yeah, I echo what was said in the chat. It is awesome to see fieldwork being done again, and especially in a socially distanced and safe way.
And
that I'd like to
answer these questions that I'm just seeing now. Sorry.
Great, no worries. With that, I'd like to hand it over to our final award winner, last but not least, Sophie Everbach, who's a graduate student in the Holbrook Lab at Harvard University. Take it away, Sophie.
Yeah, thanks. Hi, I'm Sophie Everbach. I'm a G1, so first year PhD student in the Organismic and Evolutionary Biology Department at Harvard in Missy Holbrook's lab, where I study kind of resource allocation in trees. Today I'm going to give you a really brief, really broad overview of the work that I'm going to be starting at Harvard Forest, and it's entitled Eastern Hemlock Root Resource Allocation Response to Hemlock Woolly Adelgid. And now if you've attended any of the forest insect session of the symposium earlier this afternoon, you will be familiar with these little fuzzies depicted here, the invasive hemlock woolly adelgid. And the hemlock woolly adelgid is causing pretty widespread mortality in eastern hemlocks in the US and in Canada. And while the literature generally agrees that needle defoliation due to the adelgid is a kind of main component in hemlock decline, above ground biomass loss doesn't seem to be kind of the full story of this mortality. So this brings up kind of the main question of my project, which is how exactly are these insects killing the hemlock? And to investigate this, I plan on kind of focusing on what is going on below ground with root resource allocation. I'm specifically interested in the question, how does the root shoot resource allocation of carbohydrates change in response to different severity levels of the hemlock woolly adelgid infestation? And on top of this, I'm interested to see if these patterns of root shoot resource allocation track across tree ontology stages specifically. What is the pattern? Are the patterns the same between seedlings and mature individuals? And to do this work, I propose kind of 2 studies, a common garden experiment and a field study. And for both studies, the hemlock woolly adelgid infestation severity will be determined based off of adelgid density, and all trees will be ranked as having none, low, medium, or high adelgid severity. The common garden experiment will consist of me planting some 60 or more hemlock seedlings in the Harvard Forest Common Garden plot, and infesting them to the different severity levels, and then letting them run under those infestation regimes for 2 years. I will then go in and core or excavate roots and analyze them through a variety of biomass metrics and starch content. The field study will apply these same methods, but to mature hemlocks in the Harvard Forest Prospect Hill area. And we will survey for pre-existing infestation severity levels rather than applying the treatment. And then we will use the same kind of root, core, biomass, and starch analyses. By applying the same methods to each study, I will hopefully be able to compare patterns of carbohydrate allocation across tree ontology stages, as well as infestation severities. And obviously this project is really large, but it will be a really good way to engage with scientists across different career paths. As a first-year PhD student myself, I will hopefully bring in kind of other people in my lab. And also it's an especially good opportunity for undergrad work, specifically through Harvard Forest's summer REU programs, and also ongoing semester work with Harvard undergrads. So thank you all, and I'm really excited to join the Harvard Forest LTER community.
Thank you. Let's give a round of applause to all of our award winners and presenters. That was really cool. Everyone did a great job, and it was just great to see the breadth of research topics that our graduate students are covering, and really just want to emphasize that this is exactly what we want to encourage with our research awards and with the graduate community as a whole, is that we are looking at all kinds of research and we want to celebrate all the types of research. With that, I think we have time for a couple questions. We've been given a little bit of extra time. So if you have any questions for the presenters or for me, please jump in.
So Luca, seeing no hands up immediately, there was a question in the chat that I think you answered, but it's worth reading out to everybody. It was a question about what the graduate student funds can be used for.
Yeah, that is a great question. I actually might toss it over to Jonathan. Broadly, as I hope our examples of our previous award winners show, we will accept a wide variety of things, but there are some specific limitations which I can't remember. So Jonathan.
Yeah, I'm almost ready to punt it to Meg, but the particularly due to COVID, there are different restrictions now than there will be in the future, hopefully. But for example, laboratory time, travel expenses, conference expenses, equipment costs, your own time. If you're putting in hours towards a project, you can get, some salary. There's a cap on the limit of it, which is escaping me at the moment. Does anybody remember that number? More coming on the rules about the Graduate Student Research Award, including details about what can be funded and how much funding we are offering. Sorry about that, I was unprepared.
That's okay. Thank you, Jonathan. I was also unprepared for that, but it will be enunciated in our official call. Do we have any other questions? Any other hands up? Okay. I see in the chat that $2,500 was the limit last year from Meg Hastings. Thank you, Meg. We have Jess has a question, has her hand up. Please go ahead, Jess.
This was awesome. You guys are so cool. I can't wait to hear about all the projects and what you guys find out. I guess my question, I asked it a bit in the chat, but just for Nikhil, I think this exit work is so cool. And I think it's so important for thinking about carbon balance. And I was just wondering if you have a sense of, like, I have no idea. So this might be very clear that everyone knows it, or maybe no one knows it, but how much is the exit per tree in terms of general carbon movement throughout the tree or carbon uptake in the leaves? Do you have a sense of this? Or does anyone have a sense of this?
There's a paper from the Duke. There's a lot of research from the Duke FACE experiment on this, and there's a paper by this guy called John Drake, who I don't know, but is pretty famous, who actually quantified like the total exit output there. So I can find that and drop it in the chat. But yeah, I don't know the number off the top of my head.
Thanks, it's so cool.
We also have, I think, a little bit of information on that in the carbon synthesis paper, where we estimate exudation flux. I can't remember what the percentage is, but it's like 5–10% of either GPP or MPP, something like that.
Wow.
Yeah.
Okay, cool.
Yeah, I
that's
remember
based
that
on
too, but not the number.
Yeah, but that's a that's a fairly sparse data set, so it's extrapolating from relatively few measurements. So I certainly think there's a lot of room there for additional work.
See, Audrey's got her hand up.
Yeah, I have a question for Sophie. This might be serving my own interest, but so I was looking at how non-structural carbohydrates are drawn down and related to mortality in oaks defoliated by gypsy moth. Is your work, would that be able to, the sampling that you're planning, would that be able to address a similar question with hemlock and hemlock woolly adelgid? I know that non-structural carbohydrates act different in conifers though.
Hopefully it will be able to do something similar. We're still early stages here, but yeah.
That would be really cool. Yeah, I was explaining my research to my husband over the weekend. He's like, you should do that with hemlock and hemlock woolly adelgid. So passing that along, yeah.
Okay, well, if you have any more questions, I'm sure that you can follow up. You can always follow up with me if you have any questions about the graduate community in general, but I'm guessing that our award winners would be happy to welcome questions, follow up after the talks. Audrey, I'll toss it back to you.
Perfect. Thanks, everybody. That was really exciting. Can't wait to find out what you discover. Before we move on to the next session, Lucy Lee is going to announce the winners of swag from the student poster session yesterday. Go ahead, Lucy.
Thanks, Audrey. And yeah, thanks to all the students who participated in the student poster session yesterday. It was really awesome, lots of great work. And so we have 3 winners. The winner of the sweatshirt is Sarah Garvey from BU. And then the winners of the hats are Emma Conrad Rooney, also from BU, and Paige Kuba from UC Davis. So I will be writing to each of you to get your mailing addresses and any other information. So congrats to everyone that participated, but congrats to the winners of these fun swag items.
And just to be clear, I believe these winners were chosen at random, so it is no indication of the quality of the others or anything. It was Lucy's random number generator.
Yes, I tried to be as fair as possible.
Awesome. All right. Yeah. And thanks again to everyone who presented posters yesterday. They were all excellent. And it was very enjoyable to chat with a lot of you. Okay. So we're going to be moving now into our last session for the symposium, which I decided to call Putting It All Together, Interacting Global Change Drivers, Broader Impacts, and Data Curation. So a few different things. The first 2 talks are going to be focusing on interacting global change drivers in the context of the Harvard Forest LTER program. And our 2 speakers will be Scott Ollinger from University of New Hampshire and Jonathan Thompson from the Harvard Forest. Scott, you are up first. Are you ready to share your screen?
Sure, can do, as long as my screen cooperates. I think that worked. Thumbs up or nod of a head or something. All right, good. Oh, I've already advanced. So yes, I'm going to talk about multiple stress effects on northeastern forests. A lot of people have contributed to this work, including the people that are named here. Many others. If you're part of this and I haven't listed your name, my apologies. What I'm going to talk about, oh, and I should also give a nod to Jack Hastings. The only nice color figure I had for the region was from his work. So that's the figure here, which I'll highlight in a few minutes. So I'm going to talk about multiple stressors on forest growth and carbon, net carbon balances then and now. We started thinking about this about 20 years ago, and we made some early predictions back then, and we've decided to revisit some of those, which has been fun. In the meanwhile, there has been uncertainty and debate about things like the physiological effects of rising CO2. We also know that air pollution has declined, ozone concentrations have come down, as well as deposition rates of nitrogen and other elements since the Clean Air Act amendments were passed. There also are some interesting contrasts in how empirical trends in forest productivity have changed in different parts of the region. We saw Audrey talk about increases in growth in oak, and Bill Munger has also shown that carbon uptake has generally increased over the years at Harvard Forest. That hasn't been true elsewhere, and we're not really sure why. So then at the end, I will briefly touch on some work that we're starting to get going. I don't really have results to show from it, but at least wanted to give an update on where we're headed. So this is work from about 20 years ago. We used a model called the PNET ecosystem model, which does carbon and nitrogen cycling in forests and soils. It includes climate and carbon and nitrogen pools in different vegetation pools. It handles litter decomposition. Litter is soil organic matter, ends up as it decomposes, drives nitrogen mineralization. There's nitrification, there's competition between plants and soil microbes, although it's not as sophisticated as what some models do today. And we've made a number of improvements to it over the years. Back when we did this work, we were interested in how ozone and rising CO2 and nitrogen deposition, as well as past land use history, had influenced the present day. By present day, I'm talking about the year 2000, rates of NPP and net ecosystem exchange. So we pulled a bunch of data sets together, and we added some algorithms to the model that it allowed it to simulate ozone effects. And these are just a few of the results. This is using the land use history of Harvard Forest as an example. Net ecosystem exchange under a baseline scenario, which means no rise in CO2, no past land use history. We just kind of flatline the model and have everything run to equilibrium, so it's at steady state. Then we add multiple perturbations, like the land use history alone without any atmospheric change that has been documented for Harvard Forest is this thin line here. The model was run from the year 1600, and I'm not showing that whole sequence, but we spun it up for a couple centuries, and there were disturbances prior to 1880. So coming into this, there's a net positive carbon balance coming into this. If you add these atmospheric effects, you get fertilization from ozone, a little bit of a fertilization effect of CO2. I'm sorry, fertilization from CO2 and nitrogen, and then some of that is drawn down because of ozone. And so we were able to kind of use a model to do interactive multi-stress experiments that are hard to do in the real world. And looking at changes in NPP, we could, for example, simulate all of these sites across the Northeast, assuming no land use history, that there had never been European colonization and never been harvesting and farming and all that, and that there were only these atmospheric effects, background atmosphere, CO2 only, so on and so forth. We redid that with all those atmospheric scenarios with a history of agriculture, which was based on the Harvard Forest history, as well as a history of timber harvesting, and we used records for Hubbard Brook to do that one. One of the interesting things that came out of this is the most disturbance intensive scenario, which is the most realistic or the most accurate for much of New England, is an agricultural land use history with all of the atmospheric effects, CO2, ozone, and deposition. And with that combination, we get a predicted NPP for the year 2000 of about 1,100 grams per meter squared per year. The least disturbance intensive scenario is if humans had never, or if Europeans had never colonized New England, we'd have a background atmosphere and no historical disturbance from logging or agriculture. And interestingly, we got about 1,100 grams per meter squared per year for that scenario also. So one of the suggestions from this was that these different global change factors have kind of equally offset one another. And that if you are looking for changes in growth in tree ring records, for example, you might not actually see it. This was just a hypothesis. We have been interested in revisiting this. There's a couple things that we have had to deal with. One of the reasons we want to revisit it now is because, as we know, ozone concentrations have come down quite a bit. These are the AOT40 is the monthly ozone index we use in the model. And it's come down quite a bit. This is from 1970 to 2020. Nitrate and sulfate deposition have both come down quite a bit more actually than what the Clean Air Act targets were. And then this is a figure from the Finzi et al. Carbon synthesis paper showing changes in climate. There also are things that I'm not addressing yet. We know there are invasive insects, which we would like to be able to tackle. There also are some unexpected trends that have unfolded. Nitrogen oligotrophication, if you've heard that one, I've learned to pronounce it. I haven't dealt with it in a model yet, but it's something we have to think about, I think. There also were uncertainties in how we simulated the effects of rising CO2. And between then and now, there has been widespread evidence that has developed that suggests that water use efficiency has been increasing in ecosystems worldwide, and certainly in the kinds of forests we have here. This is a figure from a paper by Trevor Keenan in 2013. He used flux tower data to estimate water use efficiency. This blue line is the Harvard Forest EMS tower, changes in NEE uptake, negative numbers for net uptake in this case. And Trevor estimated by inverting a model against this data record that a lot of the change in instantaneous water use efficiency has been due to a change in stomatal conductance, abbreviated with a GS, G subscript S, physiologists like to abbreviate conductance with, more so than a photosynthetic enhancement. And water use efficiency is the ratio of evapotranspiration, or it's the ratio of carbon gain to water loss. Instantaneous water use efficiency is estimated by the ratio of photosynthesis to stomatal conductance. And using conductance takes away the effects of things like atmospheric humidity. So it's kind of an intrinsic metric of how ecosystems deal with carbon and water. That matters for modeling because we had used an approach that assumed a constant CI-CA ratio. That's the ratio of leaf internal CO2 to ambient CO2. And we used that ratio, the idea that that ratio would remain constant, to estimate an increase in internal CO2 as the partial pressure of CO2 in the atmosphere goes up. So we simulate an increase in photosynthesis as well as a slight decrease in stomatal conductance, which is not what this showed here. Other studies have been kind of all over the place. A postdoc in my lab a couple of years ago decided to try her own luck at resolving this. This is work by Rosella Guerrieri, who some of you know from her work at Harvard Forest, coring trees. We used a dual isotope approach in tree rings. We sampled 8 sites across the US, these red dots over here, one really dry site out in Flagstaff, Arizona, ponderosa pine site. And the logic of using dual isotope C13 as well as O18 is that we've heard other people mention that C13 is an indicator, C13 in plant tissues is a good indicator of water use efficiency, but it can't be used to separate whether that's because of an increase in photosynthesis or a reduction in stomatal conductance. O18 is only sensitive to transpiration. So if you measure both of those in plant tissues, it allows you to separate those two factors. And this was a ton of work. I'm summarizing it very, very quickly, but Rosella took thin sectioned the cellulose parts of tree rings and analyzed them 30 years worth for 2 species at each of these sites. And the results were pretty consistent. They were actually quite consistent. All the sites did show the increase in water use efficiency, including Harvard Forest, that other people had reported. Not as much of an increase in water use efficiency as you get if you look at flux tower data. We can talk about why that may or may not be. But interestingly, this is the kind of important thing here, is that the estimate of internal and ambient CO2 is ambient CO2, that's not an estimate. As ambient CO2 has gone up, the estimated internal CO2 has also gone up, which suggests widespread photosynthetic enhancement being at least part of the change in water use efficiency. And it turns out across all of these sites and species, the photosynthetic enhancement was widespread. The only sites that didn't show dramatic or at least a significant enhancement of photosynthesis were sites that are frequently drought stressed, like the Ponderosa Pine site in Arizona, which the isotopic evidence suggests that the trees there are more relying on a reduction in stomatal conductance. So this generally supported the approach, the constant CI/CA ratio approach that we had been using. We have modified our original CO2 algorithm, was a little bit strong-headed as far as fertilization effects, and we've used more recent data for that. Using more recent CO2, ozone, and nitrogen deposition data from Harvard Forest and nearby sites, we've done some new, and by new I mean like yesterday. These are new graphs. I was talking to Bill and a few about this just in the last couple of days. We did the same structure for the model simulations, running the model from 1600, in this case, to 2020. Those graphs get really messy, so what I've done is just reported here the average predictions for 3 time periods: 70 to 75, 90 to 95, and 2010 to 2015. And this is predicted NPP in grams per meter squared pure of carbon, and this is predicted NEE for these 3 time periods. And these scenarios down here, no land use history is, we use real climate data, at least for the period that we have real climate data. So there's climate change affecting these predictions, but not a land use history. This scenario has land use history as well as climate change. Again, we've added CO2, we've added ozone, and we've added nitrogen, and then all of these factors together. I'm probably going to run out of time, so I'll go through this somewhat quickly. One of the things that's interesting is even without land use history or CO2, the model predicts a pretty substantial increase in growth over this time period, and that would be the model response to the change in climate in the climate data inputs. So the effect of climate change on its own, the model anyway thinks is fairly substantial. There is an additional and also fairly substantial effect of CO2. Land use history, I haven't said this much, but the effect of an agricultural land use history is to leave soils depleted in nitrogen. So the land use history effect, you end up with, when you include that disturbance history, you end up with lower NPP, which comes back when you add CO2. And I won't step through all of this. This is NPP and this is NEE. We never get NEE that's as high as what the EMS tower reports for reasons I still am not clear on. I guess one thing I'll say, just to summarize this, is the ozone and nitrogen in the older analysis kind of equally offset one another. And that's still true in the case of NPP. In the case of NEE, nitrogen and climate both have effects on soil respiration, where ozone doesn't have much of an effect on soil respiration. So the responses are a little bit different. But the ozone effect since 1970, the reduction in ozone since 1970 has caused, in terms of the model predictions, about a 50 gram per meter squared per year increase in net carbon exchange over what it would have been if ozone concentrations hadn't declined as much as they have. So a couple of things, I'll go through this quickly. This is not my own work. I'm just going to summarize some things that have happened outside of Harvard Forest that might be of interest to people. We have a somewhat longish record of wood growth and kind of NPP at the Bartlett Experimental Forest. We don't see increases in NPP over time. This is only since 2005. This is work from Andy Wimet. But there has, in that time period, been a significant reduction in at least wood growth, which we don't have litter fall and root growth for all of these years, but wood growth is a dominant fraction of that. So it's wood growth there is kind of declined by about the same amount that this is again from the Finzi et al. Carbon synthesis, about by the same amount that it's increased at Harvard Forest. Not really sure why. These blue dots are a more recent record from a site in Durham, New Hampshire called the Thompson Farm. And the reason I'm including this, we've had two really big major droughts in the last 5 years. This is 2016, and then just this past summer. One of the things that was interesting, the Bartlett site got hit really hard the year of the drought, whereas the Thompson Farm site got hit hard the next year, probably because of ring porous oaks. Maybe the growth strategy of oaks led to that 1 year lag, I'm not sure. In the 2020 drought, the trees at Bartlett didn't seem to mind the dry weather, but the trees at Thompson Farm did. And we'll have to wait till next year to see if Bartlett has a 1 year lag in that case. So it's kind of an interesting puzzle that we're not sure how to explain. There's also nitrogen oligotrophication is something I think we're going to hear more about. People at Hubbard Brook used to be bent out of shape that there was too much nitrogen, and now they're bent out of shape that there's not enough nitrogen. But I'm saying that somewhat tongue in cheek, that there really is good evidence that nitrogen cycling rates have been declining there for reasons that can't be explained just by the decline in nitrogen deposition. This is from a paper by Peter Grofman, who's been measuring things like the mineralization rate, forest floor CN ratios, N2O fluxes, and for as long as they have been measured at Hubbard Brook, those nitrogen cycling parameters have been declining, and the growth rates have also been declining. I don't have data from Hubbard Brook to go along with what I showed for Bartlett, and it's not just at Hubbard Brook. Don't try to read this. This is from Peter's paper. It's a schematic. He's implicated rising CO2, pumping carbon into soils, which he suggested might be widening the CN ratio and slowing down the decay rate. This is work from the Bartlett Flux Tower, where we've seen an effect of the vernal window, or basically when soil thaws. If the soils thaw early in the growing season, the Flux Tower in the subsequent summer, we have a lower maximum photosynthetic capacity the subsequent summer. So there may be some effect of microbial activity during the mud season, basically, maybe losing nitrogen early in that season before plant activity has kicked in. We're not
Scott,
sure.
You're at 18 minutes, just ahead.
Okay, great. I'm wrapping up. So a couple of other papers. It's not just in the Northeast. These are just a few other papers that have talked about this. And then just very quickly, what we're planning on doing, I don't have any results to show, but we're in the process of integrating the version of PNET that does nitrogen cycling into the Landis model. And you've heard about Landis already, so I don't have to go into that, but Landis does a lot of things that PNET doesn't, disturbance and species change over time. Landis already uses PNET for estimating growth so that it can deal with competition, but it doesn't have nitrogen cycling feedbacks, which we think might be important. And then lastly, this is where Jack's work comes in. We've always wanted to have an estimate of leaf nitrogen when we do simulations over the region, and that's been difficult because we really would like to have hyperspectral remote sensing data, which we don't have, but we've been working with broadband remote sensing data based on data we have that show a significant response in the NIR region. So this is a preliminary map of canopy nitrogen and using AMAX nitrogen relationships that we've measured. This is work from Jack Hastings, who also has a preliminary map of maximum photosynthetic capacity. I think that's all I have. It is. Sorry if I went a few minutes over. I'm happy to take questions. And thank you, Audrey and Jonathan.
Yeah, this is very cool. I think we have time for one quick question, if there is one.
There's a question in the chat about FUF. in the
Yeah.
figure. That's the Flagstaff unmanaged forest, and that's the Ponderosa, the dry Ponderosa site in Arizona.
Cool.
That's an easy one.
Excellent. All right. Thank you so much, Scott. And we'll have time for more discussion at the end of this session. Next up, I would like to introduce Jonathan Thompson, who will be talking about more interacting global change drivers.
Oops. Here we go. You can see my screen? Great. So I'm going to continue the theme of interacting global change drivers. The question posed in LTER 6, what are the relative and interactive effects of climate change, invasive insects, and land use on the underlying forest template? I'm going to start this with a couple of interesting slides from Neil Peterson's group. Now, I should confess that he sent these to me just a couple days ago, and so I will point to him if there are questions at the end of the talk. But it's pretty suggestive of some interactions coming between climate change and an invasive insect, hemlock woolly adelgid. So Neil's group in the 90s studied two stands where in this, what I think is just a beautiful figure, where you can see the stems and the size of the stems and the origin year of those stems. They're color-coded by species. And then he returned to those stands in the 2010s, and you can see those Jolly Roger pirate flags there indicate where the hemlocks had died due to hemlock woolly adelgid. And what they found was, from examination of the tree rings, is reduced drought stress for the surviving sweet birch, black birch, and hemlock after the hemlock woolly adelgid. And that's interesting. That's consistent with what you see in experiments. It's essentially a thinning effect. It released the surviving trees, and there was quite a lot of hemlock release of the small hemlocks. But this coincides with a period of general wetting. And so is the release of drought stress that he observed in the black birch and hemlock a function of the long-term climate trends, or is it a function of the woolly adelgid, or is it partially an effect of both? And so they aim to untangle that in the next few years, and I will point to him to answer any questions about this. Now I'm going to move on to work that my lab, in collaboration with a lot of others, has been working on to address the overarching question from the proposal and the three sub-questions. And I'm going to use this as sort of an outline to guide me through the rest of the talk. I'm going to start with this first sub-question. Will the land use response to climate change and invasive insects have ecological impacts that exceed the direct impacts of these GCDs? So what do we mean by that? Adrian actually teed up this talk, I appreciate that, in the chat earlier when he asked, are people salvage logging in response to emerald ash borer? And so that's exactly what we've been looking at. So here, will the land use response to invasive insects have ecological impacts that exceed the direct effects? That was the overarching hypothesis of a CNH grant I got in collaboration with the group at UMass, with Marla Markowski-Lindsay and Brett Butler and others, and they led the social science work that started with a survey of private landowners throughout the Connecticut River Valley from northern Vermont, New Hampshire, down to Long Island Sound, where we sent 1,100, 1,200 surveys and got about 700 back. And of those, 84% of the private landowners surveyed said they would harvest in response to at least one of the hypothetical insect scenarios we presented in like a choice experiment. So let's break that down a little bit. This was the CNH part, but it combines with the LTER research shortly. Here are the factors of the responses that were most associated with the intent to harvest. You can see much of the drivers to harvest had to do with how much of the landowner's forest died. So if more than 50% died, it was almost, the odds ratio were almost 3 times over if it was just 10%, and even higher for 90%. Also having a positive influence on the decision to harvest was whether the land was owned primarily for timber production or had commercially harvested in the past. And there was a positive association with landowners who owned more forest than was small. So using this information, John Holt, who is a superstar PhD student down at Duke, he used to be up at Dartmouth in Mark Borsick's lab, but when Borsick moved down to Duke, they all went down there. He took those data and created landowner functional types. And you can think of these as a bit like plant functional types, in so much as they each have their own probability distribution of, well, how they'll react to invasive insects based on characteristics of the landowner and the landscape setting for which we knew where the surveys were taken, and we could extract information. Things like percent forest of a town had a positive influence on the decision to harvest. And he determined in this nice paper in People and Nature that there were 3 different functional types. One, 29% of the landscape falls into this always harvest category, whereas a bug shows up on that landscape, they're going to cut. They were going to cut anyway, and the bug is going to motivate them to cut now. 8% of the land falls into this never harvest category, where they could have not a standing live tree left and they still wouldn't harvest. But 63% were like swing voters. Like, it depended on the characteristics of the insect and the mortality event. And you can see there on the map on the right, the spatial heterogeneity in the valley, where a lot more always harvest folks up north. And as you move south in the valley, you get more into the lower probability of harvest. But now this was all about generic insects in hypothetical situations. Meg McLean took John's characterizations of AFTs, and she applied this to the case of the emerald ash borer in still kind of a hypothetical situation, but used a imputation map of all of the Connecticut River Valley and a parcel map, and determined that 25% of the forested parcels in the watershed may be harvested, assuming EAB was everywhere instantly, which isn't too far from the truth. It's moving very rapidly. Now, here's where we had to make some assumptions. If you just assume typical harvest behavior, EAB-induced harvest would remove 12% of the total biomass in a decade, and 81% of that would be non-ash. And so I've always found this kind of an interesting phenomenon. I refer to it as bycatch, but we got a lot of pushback from the reviewers, so we don't call it that anymore. But ash never makes up the majority of a biomass in a stand. It's, I think, on average about 8% when it is present. It costs a lot of money to move all the logging equipment up into a stand, and so it's rare that you would just take the host species. So EAB has this sort of nefarious way of killing oaks and pines, in so much as when you go in to salvage log your ash, you'll take the EAB, you'll take those species with it. And so that was an interesting application of the survey data brought into an analysis of a real insect scenario. If we had been doing things maybe logically and in order, we would have started with this study, which we actually just submitted this week. While we couldn't look at how EAB was going to impact the Connecticut Valley because it just got there, EAB has been in the Midwest since 2002. And so John Holt, using a sort of a quasi-experimental design, used the spread of EAB determined from APHIS records with so APHIS reports when the EAB arrives in a county, and then we use the FIA data to look at the harvest patterns. And so this is an actual empirical answer to Adrian's question. And what we found was, first, that EAB had a negative impact on harvest probability. And that was kind of surprising and remains a sort of hard pill for me to swallow based on the response to our survey. So I'll put a bunch of caveats around it. For example, the APHIS data says when it shows up in a county, and the FIA data is remeasured every 5 to 10 years. So putting a timing on the harvest versus the presence of EAB is a little difficult. And so those results were really marginally significant. What we did see was a real positive impact on harvest intensity. Overall, more biomass was taken in the areas where EAB was present. And what you'll see on the bottom in those box and whisker diagrams is that it actually lowered the average harvest diameter compared to sites where there was no EAB. And this is a little counterintuitive. And of course, we don't know exactly why, but it's just possible but that a salvage operation opens up the the silvicultural options, and they end up taking more. And they see that both lowered the diameter of both ash and non-ash species, my bycatch, so to speak. So as far as next steps on this particular research is, we're going to integrate the EAB and hemlock woolly adelgid into the Landis model so that we can combine it with climate change effects and management to take a better look at how that might work. And then Dave referred to this grant that he got, he led and with the group at UMass again, to survey foresters and loggers, because that's important. At first, we just interviewed the landowners, but it's really foresters and loggers who are making the decisions about what is actually taken. And Dave is already has a lot of sites measured with Tony D'Amato, who's involved in this research as well. And we're going to look at the impacts of EAB affected stands and EAB and logged stands. And then there'll be some stand level modeling as well. That picture on the right, by the way, is one of our Bullard fellows, Bob Saul, standing next to a load of EAB affected ash. If you look close, you can even see the EAB galleries on the logs right in front. And that's just in Amherst, just down the road from where I'm sitting right now. Okay, I'm about 9 minutes in by my clock, I think. So the next question is, what feedbacks to the climate system are associated with alternative land use regimes? So one way is to look at the carbon emissions and carbon budget of New England forests under different land use scenarios. You've heard a few times today about these scenarios that we developed throughout LTER 5, working with stakeholders from throughout New England. And in LTER 5, we were really focused on both eliciting those scenarios and then simulating them with land cover change models. In LTER 6, we've been able to focus on the ecological impacts of those scenarios, including the carbon consequences of these land use futures. And this study, again, was led by Meg McLean, who was a postdoc at Harvard Forest and is now a new professor at UMass, with a lot of leadership from Matthew Duvenek, who's also was a former postdoc in the lab. And here we're using the Landis 2 with PNET that you just heard Scott describe. I won't go into it, the model too much. In fact, I'm going to stay at a pretty high level for the next few slides. I can get into it more, but I want to say two things about these. The go it alone scenario, that's a scenario with a lot of biomass harvest. It assumes a sort of everybody's in it for themselves scenario. America's really great in this scenario, so to speak. Over on the growing global scenario, here we imagine a great population influx into New England, and it is all that people can do to keep up with the land use change. I refer to just those two because I'm going to point at them later. We incorporate those into the scenarios. And I don't expect you to take too much out of these maps because they're so small, but I do want to show you up in the top right, you can see this is the recent trends. Here we just assume we keep doing for the next 50 years what we've been doing for the past 30 years. So what does that mean? A lot of red up in the north, that's above ground carbon removed from timber harvest, and some red down in the Boston area with above ground carbon removed from development. And then you can see how these scenarios differ. Here's that growing global scenario with all the removal for development. And here's all that biomass energy removal from harvest in the go it alone. I'm just going to show this type of graph for the recent trends. We have it for all of them, of course. But what you're looking at first, what's nice about a model is that you can run it in a factorial design. So this top line here, the solid line, shows the landscape potential. There's no land use turned on. It's the grow only scenario. The dashed version of it above there is if you include climate change. So that you can see you get this fertilization effect, largely driven by Peanut's assumptions about CO2 that you just heard Scott talk about. If you look at the effect of all land use, that will reduce the growth by 16%. So that's the land use effect. Of that, 12% is from timber harvest and 4% is from development. This is the recent trends model. This is just the removal of above ground carbon. So this reflects just tree growth as it continues to recover from colonial era land use. So another way to look at that is the full carbon balance. And so here's at the New England scale, each one of the scenarios here. When you look to the left, you get green. That's the dark green is the live above ground carbon. That's what you just saw in that spaghetti graph before. This sort of, I don't know, olivey color. That's the wood that is either stored in, tables and 2x4s, durable goods, or is in landfills, or is still as slash on the site, but not yet moved into the atmosphere. On this side of the ledger are the emissions. The dark orange, those are forestry related emissions, and then the yellow is development related emissions. You can see growing global is the one with the most development, though it still pales in comparison to forestry. A couple of things I want you to notice. We still remain a strong carbon sink under the recent trends, even with this level of emissions. The growing global scenario is about a wash. It's in fact still a carbon sink, but just barely. The go it alone scenario, New England shifts from a sink to a source of carbon to the atmosphere, largely related to all that biomass energy. The next step for this is to look at the land use scenario impacts on albedo, and we're working with Charlotte Levy and Crystal Schaff's lab to do that. And that's work that's just taking off, but we'll be able to look at it not just from the land cover impacts, but also through the changes in species composition, as Landis is explicit about the tree species. And so we're teamed up with them to work on that analysis. I'm running out of time. The last question is, how do interacting global change drivers affect natural resource decision-making at sub-regional scales? I won't go too far into it, but it's been very gratifying that all of this modeling infrastructure was used as we worked with the state to write the land sector report, which informed the Massachusetts decarbonization roadmap. Governor Baker has committed the state to reach net zero emissions by 2050. And so we quantified using the LTER models, quantified the land use contribution to the state's emissions, and are now working with them to team up with other New England states to find sequestration opportunities as they left themselves up to 15% of their emissions, which they're able to achieve through sequestration, carbon offsets or other types. So we're still working with the state and Harvard Law School, in fact, to try to identify ways to do that. That's all I have. I want to say that I collaborate with tons of amazing people on this research, and I've listed them on the right, and I think that's it. Thank you very much.
Thanks, Jonathan. That was really a lot of super cool work. We have about a minute for any quick questions. Yeah.
I've not been looking at the chat at all, so if there are things I'm supposed to see, I haven't seen them.
So there were a lot of interesting observations and discussion going on in the chat. So I encourage anybody who was participating in that to please share it with the rest of the group. All right, I'll do. There was a question about harvests by forest landowners and the difference between the northern and southern portions of New England with propensity to take the ash or not.
I'm not sure I understand the question. I'm sorry.
Because
did
I tried to summarize it from memory. I have to go back and read it.
Hi, I think I was in most of those. So Lucy pointed out there seemed to be a higher probability of harvest in the north that we predicted using our survey, and then additionally within the EAB study. And I answered with, yeah, the higher probabilities were higher in the north, and they're actually higher in general in New Hampshire. Some of that came apart across state lines, which was interesting with our respondents. But then a lot of the more northernly sections was actually due to they just have higher or larger parcels. And so the parcel size was the main driver for that kind of northern higher probability.
Cool. All right. So thanks again, Jonathan. And so now we're going to be broadening out, kind of literally. Clarisse Hart will give our next presentation, and she has a daunting task of going through the broader impacts in the Harvard Forest LTER in 20 minutes. And it's especially daunting because unlike a lot of research programs, you know, the Harvard Forest does a really thorough and broad and deep job with education and outreach in the LTER program. And so with that, I'll let Clarisse take that away.
Thanks, Audrey. So I'm Clarisse Hart, and I'm the Director of Outreach and Education here at the Forest. And I'm going to be talking about a few highlights of broader impacts. As Audrey just said, there's a lot that we do and a lot of people that work on this. So I'm representing many people's work, and this is definitely not a comprehensive look. You can download my slides if you'd like to. I just put the link in the chat. I'll put it up again at the end. A few of the focal audiences that I will be talking about, we have many audiences here at the Forest, but I'll be talking about students and educators and stakeholders and decision makers. I won't get to talk about the museum and tweeting trees and lots of other cool things, but we have lots of good things to talk about. Okay, so within the many projects that we do, there are some goals that kind of unite across all of them. These are strategic goals that we're really trying to work on. So if you have ideas, especially in the realm of these goals, I'm totally happy to hear them. So the first is inviting and amplifying new voices into our work and making data accessible and relevant. We can do the best long-term research in the world, but if nobody can hear about it or use it, what are we really doing? Especially during COVID, it's been really important to build virtual pathways to the work. And the last thing is engaging with long-term. Definitely, we do long-term research, but it's really important also that we engage audiences deeply and over a period of years so they can really become a part of our mission. So in terms of education and outreach, again, many things that we do. I'm going to focus on K-12 and our summer research program. We do also courses and field tours, which were virtual this year, and programs for grad students and postdocs, and we have Bullard fellowships for mid-career people. And I'm happy to answer any questions about that stuff. But let's dig into K-12. So the School Year Ecology program has been going for 17 years now, and down in the bottom of this slide in the blue shirt, that's Pamela Snow, who coordinates this program and has since the beginning. It focuses mainly on teacher professional development and kind of a train the trainer model. And our goal is to introduce teachers to the idea of collecting field data with their students in their own schoolyards, so something that's relevant and local to them, and then to analyze those data to tell a story. And Pamela and the scientists that work on this project engage many, many students per year. You can see a map of all the schools that have submitted data over the last 17 years. And so if we bring up these uniting goals again, I'll just highlight a few features. One is hot off the presses. It's a new partnership to broaden participation, and it's with Mass Audubon. They have said they want to start collecting some of our field data with students at 7 of their sanctuaries across the state, including in Boston and Worcester, which is really great. It's been difficult for us to reach urban audiences with our ecological field projects. So this is pretty exciting. And then you also heard from Amanda Suzzi, an LTER graduate student from UMass, who's working with us on a watershed ecosystem services learning tool to make sure that it's accessible, especially to the students in the greater Lowell region who are living right in that kind of focal area for that teaching tool. Relevant data, we are so fortunate to have an amazing information manager here at Harvard Forest who really sets our schoolyard ecology program apart from others in the LTER network, because over the past 15 years or so, he's built this incredible schoolyard database that allows teachers and students to submit all of their field data online and then also, download it, compare across sites. It's all freely available online. And there's also a graphing system where they can not only graph their own data, but do these comparative graphs across sites. He builds new features every year. It's really amazing. Our focus on data also has made us pretty successful at working on this project called Data Nuggets, which takes real ecological data sets, kind of distills them down, and we create a learning teaching tool that goes up on a national website and is freely available and widely used to make Harvard Forest data accessible to many, many students around the country and probably around the world. So we've built a couple of those and have a couple more in the works right now. And thanks to all the scientists who have worked with teachers and graduate students to make these happen. And there's a link in my talk if you want to explore them. This is a local teacher, Alicia Andrews, who did a tree ring data nugget based on her fieldwork with Neil Peterson a couple of years ago. And she was an RET, which is research experience for teachers, which is an NSF grant that allows teachers to have those really deep summer experiences. Virtual Pathways, Pamela has been heroic this year to try to support teachers with their virtual learning that they've had to do with students. She has supported 12 workshops for teachers over this academic year to just give them what they need, working with scientists to do presentations, talking to teachers about how they're doing remote learning and helping them to learn from each other. Teachers also share their lesson plans online, so we have this really robust set of resources that's been developed over the years by teachers for teachers. And then the last thing, long-term engagement. The schoolyard ecology program really depends on the long-term engagement of its staff. So we have Pamela here on the right, on the top, and then John O'Keefe, who's an ecologist who's been there since the beginning, 17 years. And then in the bottom left, that's Betsy Colburn and Dave Orwig. They've also been involved since the very beginning, working with teachers, building field protocols. And then we have relative newcomers to the program. We've only been doing it for like 5 to 8 years. That's Audrey Barker-Plotkin and Greta Van Scoy. And we've also had Josh Blazinski and Ann Lewis and many other people getting involved with the schoolyard ecology project program. So thank you to all of the scientists. And we also welcome new scientists who want to get involved. The teachers also stay involved over the long term. This is holding the certificate is Maria Blewett, who is getting one of the certificates that Pam gives out for 5, 10, and 15-year service awards. And we have given out literally dozens of these. So teachers stay engaged and they develop these data sets that, range 5, 10, 15 years. And when they retire, this is an example of a data set from Elaine Senecal, who retired after 7 years and then made sure that the teacher who followed after her picked up her data set and kept going with it. So teachers are really committed to this, and so are the students. Okay, pivoting to the summer research program, which many of you have been mentors in and know about. This is an 11-week paid internship that's been going for probably 30 years at this point. We have new program directors. Audrey Barker-Plotkin and Sydney Record are doing a fantastic job building on the incredible success that this program has had in the past and developing new things. One of the successes that they are continuing to build on is the incorporation of new voices. So here's a really great graph that shows, if you look at the bottom line, that blue line, the percentage of participants in the program who come from traditionally underrepresented groups, racial groups. From 1993 at about 10% of the program to now it's about 50% of the program. So this is due to really strong recruitment efforts and relationship building that continues. So thank you for that, Audrey and Sydney and all the mentors. making sure that the data is relevant and understood by the scientists, by the students. Sydney Record especially has been really active in leading our workshops and others for the students, so they really know how to analyze their data. The program also focuses on the complete research process, so it's not just collecting and not just even analyzing the data. It's also like, how do you do metadata? How do you enter it into the sample archive and the data archive? This is all really important, and we introduce students to that in the program. And then, of course, encourage presentations and publications. There's Audrey standing with one of her students, giving a poster. Virtual Pathways, they are doing this like gangbusters this year because it's an all virtual program this summer. So stay tuned on how that goes. There's also, they've developed a kind of distributed mentorship model where some of the mentors are based here at Harvard Forest or in the Northeast, and some of them are around the US, maybe former students who are now ecologists. That's building diversity in the mentorship pool and also just building students' networks geographically. Long-term engagement, we stay connected in a lot of ways with our alums, and mentors especially stay connected with their students. We make sure that students are connected to ecological networks, so like not just LTER, but NEON and OBFS and Ameriflux, so that they really stay in the ecology pipeline and have a lot of resources. And then the last thing is we've been tracking their outcomes in terms of their educational paths and career paths. Here's a great graph from, or diagram from a publication showing how our alumni kind of stay engaged in the environmental world, both as students and in their careers. Okay, I know I'm talking really fast. Sorry, I'm trying to get through a lot. Stakeholders and decision makers. I'm going to highlight 2 projects here. That is not to say that the other projects are not important. They are vast and important, and maybe next year I'll get to focus on those. So we'll start first with the New England Landscape Futures project. You've heard from both Lucy and Jonathan about kind of the development of the scenarios from this, which was done with over 100 stakeholders from around New England. Some of the outcomes from this, one major one is a web-based explorer, which you can find at newenglandlandscapes.org. It's this really fantastic interactive mapping program where you can explore the land cover changes from the different scenarios. And some of the outcomes from that tool, and thanks to Lucy Lee for helping me to pull this together. She's been sort of the outreach person for this project. Some of the ways that they've engaged new voices is by holding workshops for the tool all over New England, and also having a competitive process to support working groups to support their goals to use the tool. So it's not just sort of like, here's this tool, have fun using it. It's like, oh, what do you want to do? How can we help you accomplish it? And Lucy provides a lot of technical support for them to do that. The data, obviously it's up on GitHub, and also to make it relevant to different audiences, Meg McLean and a lot of our schoolyard ecology teachers have built lesson plans around that tool. To make sure that people are connected virtually, Lucy has been holding lunch chats about the tool to build community about it and to answer questions, and she's developed a really great help site with videos and an FAQ and all kinds of resources to make sure that people can really use it. Long-term engagement, because of these working groups, the tool is now being used in a lot of plans that are being sort of implemented over the long term, like the tool is now being used in Massachusetts' Healthy Soils Plan. And also there are plans to develop the tool further so that it can really meet stakeholder needs. So, whether that means adding new data sets or focusing in on particular ecosystem services that the tool is modeling. I know the Thompson lab is really interested in building that out. Okay, last thing I want to highlight is the public engagement with science at LTER's project. A lot of really great thinkers from both Hubbard Brook and Harvard Forest and the Kellogg Biological Station have contributed to this. It's a grant, an NSF grant that's now coming to an end, although they just applied for a new one in January. Some of the results from the initial kind of social science research that they did, which was looking at LTR scientists' goals for public engagement, was to show that scientists, at least at Hubbard Brook and Harvard Forest, really prioritize policy goals. And like to engage with science communication professionals, which I like to see. And then they've also built an evidence-based public engagement toolkit. If we look to the grant that they've written, which will hopefully get funded for the next kind of suite of work, is to develop this much further into the whole LTER network and develop within the LTER network and support a community of practice of not just communications folks, but information managers and educators and scientists and graduate students and all kinds of people who can really contribute to effective engagement. So that'll be exciting to have new voices be part of that conversation. They're going to do more social science research, thinking about shared engagement strategies from both the perspective of the scientists and also the stakeholders, like where those goals kind of intersect and how you can really build constructive plans that serve all the audiences involved. Virtual pathways, I know that they're planning on having that community of practice lead trainings and provide some online resources. And over the long term, they would like to help LTER sites be able to do strategic engagement planning, because it's really easy to just kind of do engagement as like a deep, but maybe kind of one-off part of one piece of research, sort of PI by PI. But if sites are really thinking holistically about how they're engaging, then it's easier to not leave out groups of stakeholders, and it's easier to really have some effective outcomes. So that's it. Thank you so much for listening, and I'm happy to answer any questions.
That was awesome, Clarisse, and you did it in less than 15 minutes. Yeah, so much going on. So I'd be, Josh, if you don't mind urging people to ask questions, I will do the same.
I encourage everybody to ask questions.
How has COVID affected the general, like, outreach, us being closed and all? How is your job and all the folks you work with really affected? And what's going to happen when we're not closed anymore?
Yeah, it's a good question. So it's taught us a lot about the virtual space, and a couple of things. One is sort of how to make the outdoors accessible to more people, which I think is a good lesson for us, because there's plenty of people for whom cost, distance, physical accessibility, doesn't allow them to come have a field tour. So I think it's been really helpful for us to start thinking about virtual tours, start thinking about ways that we can engage people differently. I will be excited when we can start bringing people to the outdoors again, because I don't think there's really any way to truly replace that. So yeah, that's my answer.
Clarisse, do you know what schools in Worcester that you're targeting? I live in Worcester, so I'm curious.
Pamela might know, but I know that it's the Broadmeadow Brook. Is that the Mass Audubon site that's in Worcester? I think it's sort of up to who they work with there.
Gotcha.
Yeah, that's true. We don't know yet the exact schools that they reach out to, but we're excited that they'll be joining us on the Budsleys and Global Warming Project and reaching some Worcester kids from somewhere in Worcester.
Audrey, you still have your hand up.
That's because I still have a question.
Yes.
I guess my question is, this is amazing work, and what are some of the, what are the biggest challenges to making all of this go, and from your experience, and how has Harvard Forest, how have we overcome those challenges to make it go?
Now, I would encourage Pamela to jump in on this. The thing that immediately comes to mind is bandwidth. That's I think the bottleneck, because there's always fantastic ideas. I think our scientists, more than other sites, just have incredible ideas and energy for outreach and education, and we like to support as much as we can, but we're only so many people. So being as strategic as we can about how to kind of take advantage of synergies and really look across projects, I think that helps us to do it.
Yeah, I would say the staff really does play into it. And I don't know if that's like related to recruitment by David Foster or anyone else, you know, along the lines of looking at people that would be outreach oriented scientists. So it's been a really outward facing kind of culture that's easy to work with. And when I sometimes am talking with people in my position from other sites, they're like, how are you getting these ecologists to do this? And same thing with, you know, I can't say enough about how much work Emery has done and how much vision he had for the projects from the very beginning. So that has really provided a structure that I think scientists find easier to work with, and has led to a deepening of what the teachers are able to do with the data that their students are collecting. And so I would say there's a lot of good teamwork going on, good collaboration across the board.
I see a couple more I saw Anne's hand up.
Luca and then Anne.
Hi, Clarisse.
I'd like to follow up with on something that Pam said. One of the things that really struck me in working with the teachers at the data workshop this year was that really, as I said in the chat, for the first time ever, they started asking questions that were cross-site questions from one school to another. And it really was only graphing data, which is easy for us, and asking those questions, which is easy for us, is a real challenge for them. And it's a challenge for them to work with their students to do it. But some of the graphing tools that Emery put up make it easy for them to start seeing that those questions exist as they start looking at their own data. And then to be able to ask that question, to try to formulate it in a way that they can answer it by using the graphing tools. And it really was a delight to see them make that leap, because it's a giant. How long's the schoolyard program been going, Pam?
17 years.
17 years. It's been 17 years, and this is the first year that they've asked those kinds of cross-site questions. It's wonderful.
Yeah,
I wish
thanks
we
for that
could do that with other people out in the public who could better understand science if they could do that.
Good point. It's definitely immersive. It's sort of, you watch it happening, like as the years build and as their confidence grows. It's great. I don't know if we have time for Luca's question.
We have time for Luca.
Cool.
Thanks, Josh. Hi, Clarisse. I have maybe a slightly broader question, but connected to things you were just saying. What advice would you give to early career researchers or graduate students about how to think of outward-facing engagement and how, like, just what kind of mindset and way to incorporate it into day-to-day work?
That is a great question. And I would probably recommend that they talk to the scientists who are doing this well, because I have all kinds of ideas about how you can build your skill set and work with different audiences, but it's easy for me to say that as somebody whose job it is to do that. So I don't know, I would kind of punt the question to, maybe Jonathan or Lucy or Anne or people who have really or Lucy, people who have done that dual role really well.
Maybe I can alter the question and say it as one or the most successful of the projects that have succeeded the most, whether they're specific schoolyard, LTER, whatever, and maybe this goes to Pam as well. What were the characteristics of the scientists that you found most successful in enabling a good outward communication?
Honestly, I think for me, it's attitude. It's, if people want to do it. And that's what I find so inspiring myself when dealing. I mean, even you guys are all hearing each other talk here. That's a form of outreach. And just the fact that people seem to really want to collaborate and really want to work with other people and to spread their message to the public, and do whatever is involved in that. For me, it's more the attitude than anything else.
Well, I think it's also being able to put yourself in a non-scientist's shoes. That's something that we don't do very often as scientists. We tend to work with other scientists and we think like scientists, and our everyday lives are sort of built around the science that we do. And most of the community out there doesn't have that background. And you have to be able to step back and look at it really from the point of view of beginning again. What were your early days in science like? And how did you grow to be who you are? And one of the things that I think is most critical in working with with everybody, I've worked with kids from, 2 on, to gray, as Clarisse puts it. And in order to learn, most people really have to manipulate things. As teachers, we tend to think we need to get through more material and go through it fast. So we want to show them how to do it and think it's done. And they actually tend to learn better if you just take the time and let them struggle through it and give them guidance here and there. And it's a really different approach than we tend to use in teaching college students and graduate students. Well, graduate students get the hands-on stuff. But when we're teaching classes to, college kids, we don't tend to do it that way.
That's a great point. Yeah, go ahead, Clarisse. I think we should
you have to
wrap up, but yeah.
I think just exactly what Anne just said is thinking about audience, like learn about different audiences, watch how other people engage with those audiences, and you'll, develop like a list of your heroes who are educators and sort of emulate what they do, especially like with specific audiences. Yeah.
All right. Well, fantastic talk, Clarisse. Thank you. And I feel like I'm saving the best for last. In our LTR site room, we're going to put you first, Emery. But I'm excited to have you round out the day because, if we all did our great ideas, but then never left evidence of it, that would not be a job well done. So Emery's going to be talking about information management. Thank you. Take it away.
Let's see, can you all see my screen? Okay, great. Thanks, Audrey. So I'd like to finish up today by talking a little bit, as Audrey said, about information management and technology at Harvard Forest. And I'm going to start with a little bit of history. So Harvard Forest is unusual as an LTR site in that our headquarters is actually at the field site. And that has some advantages. It's great for doing field work, for example, but it also has presented some challenges over the years. And one of those is trying to be able to provide the kind of cyber infrastructure support at the Harvard Forest that you would find, for example, at a university campus. And so this has kept us engaged for many years and will into the future too. It's an ongoing process. I think we've been able to catch up pretty well, but as you can imagine, the bar is always rising too. So for example, these show snapshots of where things have stood at the beginning and the midpoint of our LTR program, and we're currently on 6.5 here at the bottom. And just this summer, we were finally able to provide broadband internet to 5 of the houses that we have on Route 32 in Petersham, which are too far from the Harvard Forest campus to actually put on the Harvard network. But fortunately, the town of Petersham has been working for many years now with a vendor to create its own optical fiber network in town, and that came to fruition this past year. So we were finally able to connect our residences to that Petersham broadband. That was becoming an issue for attracting researchers to come and work here at the Harvard Forest because people now expect to get broadband even if we're out in the woods. Another little bit of history is the evolution of cultural attitudes, and this has been really interesting to see how scientists' views about sharing data have evolved over the course of our LTR program. It may surprise some of our younger scientists to learn that in the early days, many of our researchers were very reluctant to share their data. And that's still true in many parts of the world. and it's still true in some scientific fields, but it's not so true any longer in ecology in the United States, and especially in the LTER program, where we've seen a complete turnaround in attitude. So by and large now, LTER scientists especially look on this favorably, and they see sharing data as something that's good for science and good for the scientists. But there's still some interesting and challenging questions, as always, around data sharing. One of them is in front of the LTER network now, and this has to do with what should be the recommended policy from the LTER network on data use. So in LTER, sites are welcome to determine their own policy, but the network has always had a recommended policy. So for this, like a lot of research networks, we look to the Creative Commons. This is a nonprofit organization that provides a number of copyright licenses that are clearly described on their website that you can easily cite and has a legal foundation. And the recommendation for LTER, I won't get down too far in the weeds here today, but the recommendation for LTER a few years ago was to go with what's called CC BY, which means you can use the data as long as you attribute it correctly. And that's the current policy for Harvard Forest. But we're seeing a number of research networks, and this includes NEON, for example, has now moved to CC0, which is essentially your data are in public domain, no restrictions. And there's some, quite a few in the LTER network that are interested in seeing us follow that path. So this is something that's currently under discussion, and something we should probably talk about as an LTER site before our site review next fall. I want to say a little bit about data management and then how we actually do it here at Harvard Forest. And some of you may have seen these slides before. I think these are points that we can all, everyone here at this meeting can appreciate. The fact that scientists build on the work of other scientists. We don't work in a vacuum. And that doing that nowadays means many times that you need to have access to the data that earlier scientists have produced. And in order for that to happen, the data has to be accessible and has to be understandable. So there are some perils here, some of which will come to mind, some may be a little more subtle. One is if you work with electronic data, you know it's really easy for it to disappear by inadvertently deleting it. So we do have to be careful about that. There's a more subtle problem though over time, which some of you may have encountered, which is if you don't give thought to storage media and file formats, over time, those things can be very difficult or impossible to read, as any of you with 5 1/4, 8-inch floppies know. It's really hard to do that now, and it will become impossible over time. So there are issues around preserving data. There are also issues around preserving the content, information content of the data. This is a more subtle point, but it's a really important one for data management. And that is that if we don't carefully document what the data are, then over time, unless you have a perfect memory, you will forget even for your own data, much less somebody else's. So what happens here is if that information content is not carefully documented and preserved, then eventually the data essentially become useless. And the way that's addressed is with something called metadata, which is data about data I'll talk about in a moment. So another unusual aspect of Harvard Forest is that we have a research project application system, which has been in place now for many years. Those of you who work here are familiar with this. And for those of you who don't, we ask anyone who'd like to work here, to do research here, to fill out an application and to update it annually for as long as your project lasts. And this is done with an online system. And it has a number of advantages for you and for us. One is it allows us to take a look at the proposed research. So we have a small committee that Audrey leads that, you know, in general, we want people to come and work at Harvard Forest, but we do like to look at what they're proposing to make sure there aren't any, you know, potential hazards or issues, or make sure that if it's a field project, that it doesn't interfere with other ongoing projects. Almost all the time, the answer is yes, no problem. But it does give us a chance to look at that. It also gives us an opportunity to kind of let others know through our website what people are doing here. So that's good for you and that's good for the forest. And from a data management standpoint, it's also good because it gives us a handle on what's going on. So we knew who should be owing us data over time if your project actually comes to fruition and you do collect data in the field. So on that point, we have endorsed the LTER network data sharing policy, which is that in general, data need to be made available to the public within 2 years of collection or publication, We do make exceptions for student thesis projects, but in general, the 2-year rule holds. And so that's what we're looking for here. So I'll say a little bit about what we actually do. So what we ask you to do, if you haven't done this before, if you have a project that where you're ready to begin to archive data, we are happy and willing to work with you to help you with that. And we ask you to fill out a metadata form, which is just a simple Word document that's on our website. And we ask for metadata of a couple types. So one is project level metadata. This is what's outlined in this metadata form, and you can see the categories here. It's pretty straightforward information about your project, and it can be updated each year. And then we also ask for information about the data files themselves. So if you have, for example, tabular data, this is a little snippet here in the upper right of data from our weather station. We need the kind of metadata that's shown here in the lower right. Basically, it's information that explains what the variables on the table are, what the measurements are, how they were measured, what the units are, and so forth. It's the things that you would want to know as a scientist if you are trying to make use of a data set. Once we receive that metadata information from you, we then take it and reformat it into something called ecological metadata language. So this is an application of XML, extensible markup language. You can see a little snippet here in the left panel. This is a text file. It's not particularly user-friendly. It's not intended to be, but it's very computer-friendly. And so the backstory here is that in the early days of LTER, the metadata for projects and data was essentially narrative. So it was the kind of thing you'd see in a paper, for example, in the methods section. And that was useful, but it's not very amenable to direct access or by computer. And so it was almost 20 years ago now, the LTER community was part of a larger effort to develop a metadata language for ecology and related scientific fields. And that came to fruition in what we're calling now EML. So this became a major effort in the LTER community. It took sites years to translate the existing metadata into this EML format. The tools that we had at the time, software tools, are pretty rudimentary. They've since gotten a lot better. This is a fairly specialized task, so it's something that we're happy to do for you, but it has some enormous advantages once you do it. As I mentioned, this format's easily read by a computer. So if you take a look, for example, at one of the web pages for individual data sets in the Harvard Forest Data Archive on our website, you see a page that looks like this. This is the page for one of David's and Audrey's long-term plots out in Prospect Hill. And so this page is actually generated directly on the fly from the EML. So when you click on that link, we don't hard code this into the web server. This information is drawn directly from the EML. And one of the advantages of that is if we want to make any changes to the formatting or the arrangement or the presentation of this information, all we need to do is change the style sheet that's used to translate the EML into HTML. Another added feature we added to the data archive a few years ago, and this was in response to a recommendation in one of our mid-tier site reviews a few years ago, was to add some tools that would help potential users preview the data so they could get some sense of what's in the data set before they actually go through the steps of downloading and looking at it more carefully. And so that resulted in these data set previews, which we post in our data archive for all of our data sets. And these are generated by an R script, which is on our website if you'd like to take a look at it. Basically, what it does is it looks at each of the data files. This is John O'Keefe's phenology project. And depending on what the data are, for example, it may do a scatter plot matrix. This is done in R. So it's plotting each variable as a function of each other variable. Many of these aren't particularly useful or meaningful, but some of them are. So it's a way of getting a quick glimpse at what's in a data set and whether it might be useful for you. We also present some tables here, summary tables of the variables. And if it's a time series data set, it does a simple time series plot. So again, none of these are finished product plots, but they're intended to give you and the user a sort of glimpse of what might be in the data set and whether it might potentially be of value to you. I don't want to get too far into the weeds here, but this is what we actually do when you give us data. So the work is done now by Julie Hall and by me, mainly by Julie. And this is the workflow that we go through when we process a data set. So without getting too much into the weeds, I wanted to note at the top here, these are the things that you as a scientist provide. The metadata form that I mentioned that has project level metadata, metadata for individual variables, and then the actual data files themselves. And then we look at these materials. We'll come back to the scientists if there are questions. So for the data files, this is usually pretty straightforward, but we may do some reformatting, and we try to make sure that the data as far as possible, are in a format that's sort of universally used. So for example, for tabular data, we use comma separated value text files, which can be read by many, many software packages. So we take the data files, we take the metadata information that you provide, we create the EML file, and we create what's called a data package. So a data package is a combination of the EML metadata file and then one or several data files. We then run that through the data preview script that I mentioned earlier. That catches a lot of problems, and if there are problems, we go back and try to fix them. We also run the data set through the EDI evaluator. Now, EDI is the Environmental Data Initiative. This is a project supported by NSF. It currently serves as the archive for LTER data and data from related projects funded by NSF. And a lot of work has gone into developing evaluation tools, which especially compare data to the metadata. So there are dozens of tests that are applied. And again, this is very helpful, helps us to catch any issues that might be either in the metadata or in the data themselves. Once the data set passes those two things, Julie will post it in our Harvard Forest Data Archive, and then she'll send a message to the scientists asking you to take a look at it. And once we have heard back from you, or if you don't answer in a week's time, we'll go ahead and upload it to the EDI repository. So one difference to note here, and this is important, is that in the Harvard Forest Data Archive, we post the latest version of a data set, but EDI is set up to keep permanent versions of every version that we submit. So each time we submit an update, for example, to a long-term project like the soil warming or hurricane project, it keeps a permanent version of that data set and assigns a DOI, or digital object identifier. And that's actually a really good thing for science, because it means if you write a paper, for example, you can cite the DOI and the user can retrieve the exact version of the data that you use for your paper. EDI is a member of a larger group called Data One, which is also funded by NSF. Data One is actually not a data repository, it's a metadata repository, and it has metadata records for close to 1 million data sets worldwide. And so the data that we have through Harvard Forest LTER goes in our data archive. It also goes in the EDI repository, and then the metadata record goes in Data One. So this helps to make our data visible to a very large community worldwide. I want to touch on a couple other things before finishing. One is the schoolyard LTER database that Clarisse had mentioned. Many LTER programs have a schoolyard program, but we're a bit unusual in that we also collect and manage the data that the schools generate and submit. And we've been doing this for many years. So the data go back, I think, to 2004 for some of the schools. So we have a very large collection of data now. You can see on the left, this is what the online interface looks like. And we do this a little differently from our regular scientific data. So as Clarisse mentioned, this is actually an online interactive database. So teachers can log in, or sometimes the kids, if they're old enough and responsible, can actually log in and enter the data into our system. We then review it, and if it looks okay, we upload it into the main database. And at that point, the schools, and in fact anyone, can download the data and make use of it. The database also supports a photograph from each site, and we do encourage schools to submit those. And as Clarisse mentioned, we've added some graphing tools as well. So as she also mentioned, our model for the schoolyard program has been to work directly with the teachers. And as Anne mentioned, and Clarisse mentioned, we work with them every year in one of our data workshops, or sometimes several workshops, on the art of data visualization. And as you know, this can be challenging even for experienced scientists, and it's very challenging for the kids and the school teachers, for many of whom this is really a new thing. They haven't done this before. And so we try to encourage at different levels. So for the younger kids, often it comes down to drawing graphs on pencil and paper, and it's good for everybody to have gone through that and know how to do it. For some of the more advanced high school students, it may be using their own software tools, which we encourage them to do and develop graphs. And then kind of in between those, we have included some graphing capability into the online system. And so this can be used to get results right away, which is often really important for the teachers. Mainly the teachers have to justify to their administration the time that they spend in this program, and having results is actually really helpful for them and for the kids. On another topic, National Science Foundation likes to see LTER informatics people engaged in research. And this is a project Aaron and I worked on for many years, Provenance in R, along with computer scientists Barbara Lerner, Margot Seltzer, and others. And I won't go into too much detail here, except to mention that these are tools that now I've shown are all available on CRAN, so you can download them anytime if you're an R user. The main tool is the one in the upper left here, RDT Lite. This is a program that actually runs your R script for you and collects information on what it's doing and stores that as what we call provenance. And then the other tools can use that provenance and do various things that we think are useful to the scientist. So if you're interested in that project, or you're interested in looking at the actual source code, just go to GitHub where everything resides and search for end-to-end provenance.
Hey, you've got about 1.5 minutes left.
Thanks, Audrey. So I'll be quick here. We've seen in recent years there's a great interest now in sensor arrays and in doing real-time data collection and visualization. So we've had a system that's been running. It's a very simple example, but it collects data from our weather station, stream gauges, snow pillow, and presents it online every 15 minutes on our website. So you can check our website to see what's happening in Harvard Forest. Same is true for the PhenoCam data that Andrew mentioned yesterday. Those images are collected every 30 minutes. And it's done through something called the Field Wireless Network, which is an extension of the Harvard University network out into the field. And this has been going since 2010. It's rather unusual. We're very lucky to have university support for this. It uses a system of radios to connect sites. The radios are getting old. There are much better radios available now. So we're just about to submit to NSF a proposal to the FSML program to see if we can upgrade the radios and extend the network in various ways. If you're interested, I'd love to talk with you about it. Finally, as Jonathan mentioned, the site review is really important to LTR and to the forest, and NSF takes data really seriously. It's a key part of the site review. So what you can do to help us if you're a researcher here is make sure that we get your data set updates for 2018. It takes us time to process the data, so sooner is better than later for that. We'd also love to update your publications. We can do that much more quickly, so we can take publication updates literally right up to the time of the site review, but again, early is better. And it really helps if you could update your RPAs, research project applications. Even if you're not doing fieldwork this summer, it still helps us to have up-to-date information. And then on our end, we will work with you to post your data sets, post your publications. We're also going to make some changes behind the scenes. I won't elaborate on those, but there are things that you probably won't notice, but a good reviewer will notice. And then finally, there are lots and lots of people here that contribute to these efforts. Most of these folks do lots of other things besides just this, but it's always a team effort. Thank you.
Thanks, that was great. So just if you're thinking, oh my gosh, we're supposed to have 15 minutes to wrap up, I checked with Jonathan and I was like, he needs like 2 minutes to wrap up. So I think if we have 1 or 2 quick questions for Emery, and then we'll go to the wrap up.
A little fatigued at the end of the second day, I think.
Yeah, I'm looking out my window and the sun is shining too. Yes.
So hearing no questions for Emery, I only need 2 minutes because I need at least enough time to first thank Audrey for all her help putting this together. And I want to thank Josh and Danelle and Lucy for helping assist in all the behind the scenes aspects of this. all the presenters who this year more than usual had to reach outside of their silos into other silos and drag other data sets and slides into their presentations, and everybody took that on with enthusiasm and like genuine curiosity to get into other people's research. It was fantastic. And I really want to thank the students for the poster sessions and the graduate students who gave their talk today. That was just fabulous. So thank you for all of that. You know, and more broadly, I want to thank David Foster and the Harvard Forest staff and the whole LTER community for entrusting me with leadership of this very special thing that we are part of, this LTER program. It is extraordinary. I came up as part of the Andrews in Oregon, and I remember looking at first Mark Harmon and then Barbara Bond, both just hero ecologists to me, and thinking what they ran there at the Andrews was something special, and it's nothing compared to what we got. And so thank you. It's a total honor. It was a great symposium. I can't wait till we can do it in person, and I hope you all get to go outside now. Have a good evening.